For decades, science has pointed to clear culprits behind Alzheimer's: the slow, destructive buildup of amyloid plaques and tau tangles suffocating healthy brain cells. But recent research indicates these plaques are an intentional immune defense mechanism. The brain is actively producing them to fight off a common mouth infection. If the brain is building a biological quarantine zone out of amyloid, we have to look at exactly what it is trying to lock away. The primary suspect is Porphyromonas gingivalis, or P. gingivalis, the exact same keystone bacterium responsible for chronic periodontitis, commonly known as gum disease. This chart compares the presence of the bacteria's toxic enzymes in the human brain. We find these toxins in up to 96% of Alzheimer's brains, and the bacterial DNA itself shows up in the spinal fluid of 70% of clinically diagnosed patients.
Alzheimer's appears to be an active, ongoing battle against a specific, highly prevalent pathogen, rather than a spontaneous accumulation of toxic proteins. P. gingivalis does not stay localized in the gums. Once established in the mouth, it actively seeks pathways to spread throughout the rest of the body. This cross-section maps the 3 distinct routes the bacteria takes to reach the brain: hitchhiking inside infected white blood cells, directly damaging the endothelial cells of the blood-brain barrier, or crawling directly up the olfactory and trigeminal nerve fibers. Upon arrival, the bacteria deploys its primary weapon, highly toxic proteases called gingipains. Here, gingipains act like molecular scissors. They systematically target healthy structural tau proteins, chopping them into pieces. Unable to hold their shape, these fragments immediately collapse and stick together, forming the dense, insoluble tangles that are a hallmark of Alzheimer's. This changes the role of amyloid beta entirely. It is an antimicrobial peptide deliberately deployed by the brain to stop the gingipain activity. To create a quarantine zone, the amyloid beta physically traps the bacteria and disrupts its cell membrane, neutralizing the immediate threat. The tragedy is that while the amyloid successfully traps the bacteria, the prolonged presence of the pathogen triggers severe chronic inflammation spanning decades. This endless, low-level biological war between the bacteria and the brain's immune response is what drives widespread neuron death. Standard broad-spectrum antibiotics fail to solve the problem. They cannot effectively clear the brain infection, and P. gingivalis easily develops a 1,000-fold resistance to them over a short period. Without the specific tools to fully clear the bacterial invader, the brain's own defense mechanism locks into a destructive chronic siege. Researchers are now testing a new class of small molecule inhibitors, like the drug COR388, designed specifically to neutralize gingipains without acting as broad antibiotics. In animal models, COR388 drops brain bacterial load by 90%, halts amyloid beta, and rescues neurons. Because it disarms toxic enzymes instead of killing the organism entirely, it avoids the rapid antibiotic resistance seen with traditional treatments. Because the root pathogen here originates as a gum disease bacteria, you already have access to proactive brain protection today. You can suppress P. gingivalis at its source through meticulous daily brushing and consistent daily flossing, treating gingivitis early, and regular professional cleanings. To recap, evidence points to Alzheimer's being driven by P. gingivalis. The infamous plaques are actually the brain's immune response to the infection. And while targeted pharmaceutical inhibitors are in development, meticulous daily oral care is your immediate first line of defense. Shifting Alzheimer's from inevitable genetic decay to a preventable infection means the power to protect your brain health is sitting on your bathroom sink.